want to thank you all for joining us for lunch and for the first one of the year, right, Erin? First lunchtime lecture of the year. Before we get started with today's speaker, I want to say that our next talk will be March 3rd, first Tuesday in March, when we will have Morgan Gostell talking about your November expedition to South Africa, right? As part of your work on Benoni. I think I got that right. Okay, so for today's lunchtime lecture, we are featuring our newest member of the research team, so Kay Hankins, who's our new seed bank conservation botanist. And I'm not going to say anything more because she's going to talk about her own history.
Sounds good. Thank you. Well, I really appreciate everyone for joining me both in person and virtually today. Again, my name is Kay Hankins. I'm the new seed bank conservation botanist here at the Fort Worth Botanic Garden and Botanical Research Institute of Texas. And I'm super excited to be talking to all of you today about conserving plant biodiversity through native seed collection and how species distribution modeling can be a really valuable technique to support this work. So first, I wanted to provide a little bit of background about me because, as Brooke mentioned, I'm one of the newer faces here at the garden. So I earned both my Bachelor's of Science and Master's of Science in biology from Sam Houston State University in 2018 and 2023, respectively. And in between earning both of those degrees and before joining the garden here, I worked for the Chicago Botanic Garden, first as a botany field technician, collecting native plant seed, conservation scale collections of native plant seed with the Chicago Botanic Garden in the Bureau of Land Management in Boise, Idaho in 2019. And I fell in love with it. So of course, immediately after, I transitioned into a position as a GIS analyst in the Mojave Desert, supporting my crew make the operational scale collections, so multi-pound collections of native plant seed for desert tortoise conservation and habitat restoration for 14 months in 2020 to 2021. And then after I graduated with my master's, I went back as a lead field botanist in Northern California and Northwestern Nevada, making, again, operational scale collections of native plant seed for the Seeds of Success program based out of Susanville, California. Leading up till now that I'm the new seed bank conservation botanist, that I started this position in January of this year. So fresh-faced, very excited. There's a lot of exciting things coming up, so definitely stay tuned for some of those further developments in the future. But without further ado, let's talk about plant biodiversity and conservation. So it may come as no surprise to most of you that global biodiversity is under pressure due to anthropomorphic activities, including the increased globalization and industrialization of the world's economy, travel, and trade. And this, of course, has directly influenced changing climate, right? As illustrated by this lovely graph of the drastically increasing annual mean temperature on the planet, right? Which exacerbates ongoing issues such as habitat loss, habitat fragmentation, where a species' once continuous native range is now fractured or splintered into many discontinuous pockets of that former larger range. Increased incidences of introductions of invasive species and pathogens. And this is just a few examples, but all of which work together to drive up increased risk of extinction. So what does this actually mean for our native plant communities? Well, they're at risk. And in fact, RGBQ's State of the World's Plants and Fungi report, released in 2023, estimates that 45% of the flowering plant species included in their study, which was using data from IUCN's Red List, 45% of those flowering plant species are at risk of extinction. due to issues that I mentioned previously, such as the introduction and proliferation of invasive species, such as Bromus tectorum or cheatgrass in the western United States, kudzu in the southeastern United States, the increased incidence and intensity of forest fires, even in habitats and ecosystems that are adapted for those fires. So you can only imagine the devastation that is wrought upon ecosystems and habitats that aren't adapted to those fire regimes. And the introduction of pathogens such as American chestnut blight that was introduced in the early 1900s that has irrevocably altered the ecosystem in the eastern United States forever. So why is this such a problem? Well, as a lot of us know, plants are a fundamental and foundational, they play a fundamental and foundational role in nearly every terrestrial ecosystem on the planet. And modern day plants are estimated to be going extinct at rates that are 500 to 1,000 times higher than the average historical background rate seen right here. And this is problematic, right? And as you can see, this figure illustrates the drastic rate of increase for vertebrates and mammals. However, those rates and their actual original historical background rate are very similar to that of plants. So it kind of illustrates kind of those drastic rates of increases that we're seeing with plants as well. 77% of undescribed plant species are likely threatened with extinction. And the more recently a species has been described, the more likely it is to be threatened. And finally, once these species are gone, so too are their genetics, their evolutionary history, their potential, and their function in the ecosystem, which, as you can see and can imagine, can oftentimes have devastating, rippling effects throughout entire ecosystems. And I want to bring attention to this because it's not just the conservation of plant biodiversity. We cannot conserve, restore, and protect native habitat for threatened and endangered fauna without also addressing plants. So some examples. There's no red cockaded woodpecker without healthy old growth longleaf pine forests, or greater sage grouse without high quality sagebrush steppe habitat. And finally, there's no desert tortoise without their preferred food sources in shade plants. So this was the focus of the seed collection that I was doing in the Mojave Desert in 2020 and 2021. And this is actually a picture that I took of a desert tortoise in one of the areas that we were collecting seed in, and one of the more diverse sites that we had multiple collection sites at for target species that were both shade plants that provided them shelter during the months where they were actually not in hibernation, and food plants that they depend on to thrive and survive. So what can we do? Well, as I kind of alluded to already, we can collect native plant seeds, right? And that's where the Seeds of Success program comes in. It was established in 2001 by the Bureau of Land Management in partnership with the Kew Millennium Seed Bank Project. And it is the first step in the National Native Plant Materials Development Program, which aims to collect native seed for use in things like research, development, germplasm conservation, and ecosystem restoration, with the overarching goal of increasing the quality and quantity of native plant materials available for restoring and supporting resilient ecosystems. So how does this process work? Well, the first step is to develop a list of target species relevant to your project or your collection goals. So are you collecting seed for conservation, ecosystem restoration and stabilization, seed bulking? all of these answers to these questions drive what kind of species you plan to collect, how large your populations need to be, and how much seed needs to be collected to actually support those goals. The next step, which is often the most fun step, in my opinion, is actually scouting for suitable wild populations and then monitoring them for seed collection. And that's where the adventure really begins, right? And that entails asking yourself a number of different questions, such as, what is our target species phenology? What kind of habitat does it prefer? Is it frequently associated with any other plant species? Are there any species that closely resemble our target? How can we reliably tell them apart? How close is the population to the road? Has it been previously sampled? From what provisional or empirical seed zones would our collections provide us the most utility, would be most useful? And are there any modifying factors at any of the sites we are visiting that may harm the population or reduce seed viability? It's a lot of factors that go into determining what kind of populations are actually suitable for collection. And at times it can seem a little overwhelming, especially when you're dealing with such large areas with which you can potentially collect those plants. So just as an example, here are some maps of the area of operations that I was working out of this past field season in Northern California and northwestern Nevada. This map here illustrates all of the available BLM land that we were permitted to collect our species targets on. And it may be a little hard to tell here, but it's a lot. The West is predominantly public land. And so there is a lot of land that we could potentially work with. However, for some of our species, we are only authorized to collect in particular provisional seed zones, particularly more arid seed zones, as we start to prioritize more arid adapted ecotypes to support future restoration efforts as our climate continues to change. And for one target species in particular, Great Basin wild rye or Lima cinereus, we were only able to collect in one empirical seed zone. And these are actually determined empirically through common garden studies to determine what areas the seed is able to actually successfully establish and grow. Right, so we were trying to target one of the more arid areas, which, as we found out, it does not like. We really searched a lot of that area, and the fact of the matter is, it is very associated with ephemeral or seasonal water sources that just did not exist in that particular target empirical seed zone, which was sad for us, but is still extremely valuable data for us to have now that we can use that information to inform how we plan on restoring Lima cinereus in areas such as this. Maybe we can't, and we definitely probably aren't going to find any suitable populations at the very least to collect that plant in the area highlighted there. So negative information is still valuable information to have in science, right? But you can see why having a technique available, such as habitat suitability modeling or species distribution modeling, might be an enticing prospect, a technique that can use where we know a plant has occurred to make predictions about where we might be able to find it, right? So regardless, scouting always involves two things: a lot of time and a lot of driving. And oftentimes, it's a scenario kind of like this one. This is a photo from a field guide to roadside wildflowers at full speed, which I really resonate with, because oftentimes when you're out in the field, you're seeing a blurry approximation of your target species through your peripheral going 70 down an interstate, right? You'd be surprised. My technician last season was pretty much endlessly amused by the fact that you could determine one of our targets just by the shade of green it was from afar on the hillside, or just the vague shape of it in the distance. So it does. Sometimes it looks a little bit like this. And it oftentimes involves driving on roads that may or may not actually exist. And so this is a picture I took as I was following a line on my map that was apparently a road that, as you can see, probably was not one. And if it was a road off a cliff. So that was a fun day. But the funny story about this misadventure is we actually were able to find a population of our target plant species by just making this wrong turn, right? So a lot of meandering, which is a lot of fun as well. So once you've done all of that and taken all your wrong turns and found your suitable populations, it's time to monitor your target population until the seeds have matured enough to collect. And for each species, this looks a little bit differently. This plant here is Asclepias speciosa. With milkweeds, it's really fun. It feels a lot like seed Christmas when you collect them. You bag the pods when they're immature, and as they're wind dispersed, you want to bag them so that you can make sure that you capture all of the seed before they spontaneously pop open and dehisce, and you lose all of the seed to the wind. Right, so that's one method of actually collecting the seed from these populations. And as we found out, it takes a really long time for these guys to mature. We were able to make this collection right before the end of our season, and we had been monitoring it for more than 8 weeks, I think, at that point. So once your seeds are actually mature, it's time to collect. And this is a picture of me with a tennis racket and a tarp on the ground, gently tousling the treetops to capture mountain mahogany, Cercocarpus ledifolius. And so there's lots of different techniques you can use to actually collect the seed, right? This is just one method. Stripping is very common with grasses. There's different techniques you can use. But what is always the same is that we collect only 20% of the available mature seed at our population in a given collecting day. And this is to reduce the impact, the overall impact upon the populations that we're collecting the seed from, while also maximizing the amount of genetic diversity that we're able to capture with our collection effort. So it's important to note that it's often tempting when you're out in the field collecting plant seed to go and target the plants that are the biggest or have the most seed. But we need to also target smaller plants with different morphological characteristics and capture those unique genetics throughout the entirety of our population, instead of just kind of targeting what is the most convenient for us at the time. So that's our overall objective. Maximize the genetic diversity of the plants in the seed that we sample. Don't harm the overall population by over collecting seed at those sites or in consecutive years. And then also collect from unique ecozones that will also further maximize the genetic diversity captured by our collection, and then subsequent collections of these species in later years. And then lastly, once our seed is collected, we place it into these lovely just paper lunch sacks that we store in a cool, dark place until it's ready to be shipped. And for me, with the Seeds of Success program, this was the full extent of my involvement with the seed collection process. But that is by no means the seed's final destination in this process. And for Seeds of Success, the next place that this seed goes is the U.S. Forest Service Bend Seed Extractory in Bend, Oregon, for further processing and cleaning. And Bend is a massive operation with, as you can see, numerous different seed cleaning machines that can handle a large quantity of seed. in a very short amount of time. So our minimally clean seed goes through one or more of these different machines to be cleaned, and comes out a lot like this, with all excess woody debris and plant material, rocks removed from the collection. And from there, the first 10,000 seeds of any SOS collection are sent to be part of the Seeds of Success National Collection, housed in the National Laboratory for Genetic Resource Preservation in Fort Collins, Colorado. This is a very large seed bank. It is a backup institution for most seed banking facilities. So that goes immediately as a conservation collection. Anything beyond that first 10,000 seeds can either be requested back by collecting institutions, or be sent to another partnering institution for further research and development. So as I said, seed collection is just the tip of the iceberg. Just to kind of give you like a rapid rundown of the full process. The next step is to conduct research and develop seed zones for the and practices for the successful cultivation of native seed in their establishment, so that we can produce stock seed to be transferred to large-scale private growers, who then grow out and bulk the seed up for large-scale restoration purposes, which is then purchased back by the federal government and stored at these massive BLM seed storage warehouses, capable of storing upwards of 800,000 pounds to 1.8 million pounds of native plant seed. And during particularly bad fire seasons, one of the figures that I saw was the BLM may purchase upwards of 7 million pounds of native plant seed in a single year for some of these larger restoration projects. So having these large warehouses storing native seed is extremely important. And then that provides a readily available supply of appropriately sourced, regionally adapted, which is really the most important part of this, regionally adapted native plant seed for use in restoration. And then the process continues, right? So now that you kind of have an idea of what's going on at a national scale in large scale operations, what are we doing for Texas native seed collection here at the Fort Worth Botanic Garden and Botanical Research Institute of Texas? Well, did you know that we have a seed bank right here in the BRIT building? And if you did know, I would reach to say maybe a lot of people haven't actually seen it, right? So I took some pictures. So this is our lovely little seed bank that we have here in the BRIT building. We're a much smaller operation, as you can see, than what the U.S. Forest Service has in Bend, but we're also dealing with much smaller quantities of seeds. So we're doing all of that processing actually by hand. So what are our main goals and objectives for native Texas plant conservation and seed collection? Conservation primarily of Texas rare and threatened native plant species, but also collaboration with private landowners, federal, state, regional, and municipal partners that help us conduct this work. Seed rescue from populations that are threatened with immediate or impending destruction. Research to develop best long-term storage practices, seed germination techniques, and seed viability testing, so we can continue to learn from and monitor how the seed that we are collecting is faring under long-term storage conditions, and how to best store those seeds on a species by species basis. And finally, develop the types of protocols and procedures that will best ensure we are able to conserve, maintain, and curate the Texas native seed collections that we do make here at BRIT. So for the remainder of this presentation, I'm going to be primarily focused on the conservation aspect of this process and our goals here at BRIT, and how species distribution modeling can be a useful tool to help support this work, right? So habitat suitability modeling, species distribution modeling, ecological niche modeling, it's a technique with many names, right? And I decided after I had submitted the title of my presentation that I was actually going to stick with habitat suitability modeling. So, you know, what can you do? But, and I'm sure people would maybe in the field would argue with me a little bit, but I'm using all of these terms interchangeably, right? Because to what degree there is a distinction between all of them, I think might be a little bit arbitrary. Talk to me later if you think differently, right? Okay. So going forward, what is habitat suitability modeling and how does it work? Well, at its core, habitat suitability modeling aims to quantify the species-environment relationship by estimating an organism's ecological requirements or its environmental niche, allowing researchers to better elucidate the factors that govern species distributions. These models are trained using a combination of binary response variables representing both a species presence in a landscape where it is known to occur, as well as species absence data, or in this case, most often the case, pseudo-absence data. Because as we found, most of our collection data is where species are, not so much where they're not, right? So we have to kind of have a way of telling our models, okay, these are where our species are known to occur, and these are areas where they're not known to be present, right? And there's a distinction there. They're not true absences. We haven't been surveying in many cases, all of the areas that we can possibly survey to definitively say these species are not here and will never be here and have never been here, right? And we use this data in conjunction with environmental predictor variables that are fitted to species response curves that are then used to fit our organism's realized niche, right? And this is then projected back into geographic space to generate our actual habitat suitability model predictions. And when we do this process, we use 80% of all of our occurrence data, our presence and pseudo-absence data for the use in training our models. And then we withhold 20% for use in model evaluation. And the purpose of this is to determine how well our models perform in classifying true absences and true presences. So there's two general types of evaluation metrics. There's threshold dependent and there's threshold independent metrics. And I won't necessarily get into all of the nitty-gritty details about each of these, but I want to just like clarify that there are two different types of metrics because they function a little bit differently, but the end result is the same. We're trying to evaluate model predictive performance by how well the models are able to classify true presences and true absences. And depending on how they function, you can see why this might be important. We want to find a threshold at which we maximize our true presences that are predicted to be present, true presences, and minimize any of the type 1, type 2 error that could occur with misclassification, having absences that are predicted to be present when they're actually not, and then have presences that are predicted to be absent when they do not in fact occur at those locations. So the cool thing about habitat suitability modeling is that we can generate predictions not just under current climatic conditions, but under future climatic conditions as well. And this uses a number of different models and also incorporates human impact on carbon emissions mitigation and socioeconomic issues that may shape our futures. So it's really, really kind of a cool technique that enables us to kind of see where are these species predicted to be under our current conditions as we have them now, and what might they look like in a couple of decades. But the truth about modeling predictions is they aren't fact, right? We're not wizards. I wish we were. But what we're doing here is not magical, although it may sometimes feel like it, right? There's no biological crystal ball that can predict the future of exactly how climate change and human activity may actually shape these species distributions and impact habitat suitability. But what they are hypotheses, and hypotheses need to be tested. This field is often criticized for the inherent degree of uncertainty that comes with generating model predictions. And I argue that the only time that we truly run astray is when we treat our hypotheses as truth, right? Because we can't make better, more accurate, reliable models unless we make those models, ground truth them, and then use what we learn to make them better, right? And I think that's a part that is oftentimes missed sometimes in some of the current literature out there. We're making models, we're drawing conclusions, and then we're not actually going out to ground truth those conclusions to make our models better. Some people are, and I'm very glad. I hope to be one of them in some of the work that I do here with rare Texas threatened and endangered plant populations. But that's a debate that is often discussed in this field of biogeography and species distribution modeling. So I'm going to breeze through some modeling theory and assumptions because it shapes how we use them when we're applying models for conservation applications. So some of those assumptions include that the species is in equilibrium or pseudo-equilibrium with their environment. And sometimes for invasive species, that may not be the case, which was the subject of my master's thesis in graduate school. The second is that all relevant predictors for quantifying a species niche are available at the appropriate spatial scale and resolution, and that your occurrence data is appropriate for your model's intended use. But why are these assumptions important, and how do they change how we model species? For our first assumption, a species like an invasive species, for example, may not yet occupy all of the suitable available habitat in its native range or its invaded range. And additionally, they may also occupy sink habitats that are suboptimal and not suitable for sustaining long-term populations. Each of these situations violate this assumption of equilibrium. The second, all relevant predictors for quantifying a species niche are available at the appropriate spatial scale and resolution. Well, missing or otherwise unavailable predictors, which inevitably there are many, we cannot possibly factor in all of the chemical, environmental, and behavioral interactions that impact species distributions. And all of these missing links result in unexplained variance in our model predictions. But we use our knowledge of a species' natural history to the best of our ability with the data that we have available to make models and their predictions as reliable as we possibly can with what we have currently, right? So we should identify these predictors prior to modeling to ensure the best possible outcome while avoiding misleading conclusions. And our occurrence data needs to be appropriate for our model's intended use. How we choose to source our occurrence data is entirely dependent upon the cost of errors of omission and commission, like I mentioned before. So predicting absences to be an area that is actually suitable when they're not, and vice versa, right? If your cost of commission is high, the user should source their occurrence data such that these type 2 errors are minimized. Whereas if the cost is low, one might not be as concerned with risk associated with overprediction. And I will note on that point, this is important if, for example, your predictions are informing how much area you want to designate as critical habitat for a species, right? If you overpredict the area that is suitable for that species, then you're potentially spending hundreds of thousands, tens of thousands, millions of dollars on area to conserve a species where it's actually not suitable for it to occur, right? So in that case, the cost is quite high. But if you're just trying to scout for new locations of undiscovered populations of species, maybe not so bad of a thing to have some overprediction going on, right? The cost is relatively low. So another important factor that I want to touch on, because this is very important for our applications as plant conservation biologists, but coping with collectors' bias and spatial autocorrelation. Right, our occurrence data usually consists of presence-only data sourced from online data repositories such as GBIF, museums, and herbaria, where maybe very little is known about the sampling effort, the collector's design, spatial bias, thus in survey efforts is very common, right? Where are these populations most accessible? Close to roads, city centers, museums. This may not reflect a species actual, the full extent of a species actual native range, and thus biases our predictions that we're generating based on that occurrence data if we were to use that alone, right? So what we actually do then to kind of counteract this is incorporate the same bias into the sampling of our pseudo-absence data as is observed in our presence data, right, during model development. And this spatial autocorrelation is this phenomenon where 2 or more of our environmental predictor variables in a statistical model are not independent from one another and exhibit a linear relationship. And this is again where our knowledge of our species, our target species, and their natural history really comes into play, because we'll use a statistical technique such as the variance inflation factor to assess predictor variables for their contribution to collinearity and remove predictors that are collinear. Well, what predictor do we remove? Well, not hopefully the one that is most relevant to our species, right? So knowing those variables that are actually relevant to our species and drive where they occur in the habitat, that's important information to have so we can make these decision calls during the modeling process. So just some popular niche models that are used today. The generalized additive model is a very simple correlative technique. Maximum entropy, maxent, and boost regression trees are both machine learning techniques that are very, very commonly used. And as I learned in the process of my master's thesis project, a really, really interesting technique of actually averaging the results, the predictions made over all of the correlative and machine learning models that you generate over the course of your study. Those can be averaged based on their model predictive performance to maximize kind of the contribution, the important things that are shared between all of those different models, right? While minimizing some of the areas in which they disagree, and thus kind of maximize model predictive performance across all of them. And I kind of want to just lastly run through this process, what this actually looks like. Xerilenta avia, not a plant. That is a terrestrial mollusk that I modeled for my master's thesis project. And you can see here the occurrences for model training and model testing in blue and orange. It is a species that is native to Europe. And what we were trying to do was determine how much suitable habitat is available here in the United States. And is it a risk here for invasion? Because that has really drastic consequences for US agriculture and food security. So taking that native range, I was able to generate a number of ensembles, like I said, averaging the model predictions generated over a number of the different models that I just mentioned, to create this kind of heat map of habitat suitability. And you can kind of see here, we had a very few number of actual confirmed occurrences of the species already introduced here in the United States. And you can see, for the most part, they do occur in areas of increased habitat suitability. And not only that, but for some reason, Detroit, Michigan, I don't know, you're a hub for invasive species, right? And what's dangerous about that, even though it may not be in the most suitable habitat, is that it's very closely adjacent to areas that are predicted to be highly suitable. And a lot of that area is fairly continuous and connected by things like interstates and airports and railroads, right? Which is actually where this species was first discovered here in the US, was at a rail yard in Detroit, Michigan. Right. And depending on what predictors you use, here I actually used human impact as a predictor. So proximity to interstates, roads, airports, hubs of commerce and travel. You can see how that changes depending on the different predictor variable sets that you use to generate your habitat suitability predictions. And lastly, this is actually the set that contained the least amount of variables, but is the only one that can be projected into the future. So you can see where some of that uncertainty kind of comes in, especially as we start talking about projecting models into the future to see how these distributions of habitat suitability may change over time. Again, not fact. Hypotheses. These give us a jumping off point of what we may be able to expect and how we may be able to concentrate our survey efforts in the future. And then that data can then be refed back into our models to make them better, right? But this is just an overall example of kind of like what that process is and what it looks like to actually generate these different predictions. So finally, I know it's a whirlwind. It's so hard for me to like try not to get into all the nitty gritty. My brain wants to go in a million different directions and I can't, right? We only have so much time. So I'm sorry I didn't focus enough on applications or even really the finer details of how modeling works, the caveats, the considerations. But it has a lot of potential for a lot of really cool applications in plant conservation biology, including scouting for new populations of rare and threatened plant species, which we've actually done here previously and found new populations. There's other similar studies where this has been successful in the past. So very exciting to be able to kind of narrow down that field of view to maybe some more targeted areas that may more highly correlate with where we have already found these species previously to occur and occur and to be present, and kind of give us a little just breadcrumbs to where we might be able to find them next. Generating threat assessments for potential introduction and spread of high-risk invasive species, which was the subject of my master's thesis research. We generated a threat assessment of over 21 potentially high-risk invasive terrestrial mollusk species here in the United States for the USDA to be able to use to inform how they approach monitoring for these species here in the future, so as to hopefully prevent them from ever becoming established and becoming really big agricultural pests and a risk for crop production here in the United States. Furthermore, we can use these techniques to estimate the available suitable habitat for a species of conservation concern to inform, like I mentioned previously, land management decisions such as how much area should be designated as critical habitat, exploring and quantifying niche differentiation between naturally hybridizing parental plant species and their progeny, and also just generating hypotheses for how habitat suitability may alter the quantity and availability of suitable habitat for our target species going into the future. And with that, I really thank you for coming on this absolute whirlwind of a journey of seed collection to uses of models in plant conservation biology. But I really appreciate you all coming today. Yes.
I just have a quick question. First of all, great talk. Thank you. It was really nice to see how your work and your presentation sort of bridges not only the collections that we get from the wild and preserve them in Natural History Museum or the but also the data that those collections produce and how it can be used to actually go back and inform how we conserve species. That was really cool. I was curious about the habitat suitability modeling and especially to hear your thoughts maybe on how you think botanic gardens can fit into or utilize data that results from habitat suitability modeling to either do threat assessments for the living collection at maybe our botanic garden here in Fort Worth, or potentially to utilize the space in the botanic garden to accommodate species that might especially be at risk based on analyses using habitat suitability modeling in the future, understanding what we do about threats from climate change.
Yeah, absolutely. Yeah, no, I think this would be a hugely valuable technique for kind of exploring all of those questions. Especially, you know, we know a lot about the natural history of these plant species that we're preserving here in the living collections. And we can use both the data that we have about the growing conditions here, where we're growing them in the garden, along with where they occur naturally in their native ranges or wherever. To kind of generate models that, okay, so we know they're able to survive and thrive in the conditions that we have currently, or maybe not, you know, but what does that mean then for the future? And we could generate models knowing about their natural history and some of the factors that are relevant to sustaining them here in the garden, right? And then project those into the future using a number of different, we could use human impact or not, how that drives carbon mitigation scenarios, how much the global temperature is rising, and having kind of like even a range of best case scenario. This is what we might be able to expect with the plants in our garden. Will these areas still be suitable to cultivate these species in our living collections, at least outdoors in the future? What does that mean kind of for how we're cultivating things in living collections in our garden? Do we prioritize sourcing our living materials from more arid areas in the future as the climate continues to warm? All of those questions can kind of be at least assessed to some degree using species distribution or habitat suitability modeling. And I think that would be a really interesting way of kind of planning for the future, or at least kind of generating hypotheses for, you know, what may or may not happen. but yeah, I think that would be a really, so Seth, if you're interested, I'll definitely look into some of that. Yes.
Yeah. I don't have a question, but I do have a comment. I love it.
Oh, thank you.
I thought the thoughtful approach that you took to all the assumptions in creating it reminds me of two quotes about use of models in science, and I'm sure you've heard about it. The first is all models are wrong, but many are useful.
Yes.
And the second is the best model of a cat is a cat.
It's fun. Oh, thank you so much. I really appreciate that. I'm glad that you mentioned that. Actually, there's a funny story. I went to a talk at the Association of Southeastern Biologists meeting I think it was last year's meeting, where their PhD project was kind of assessing the black box of how machine learning models are arriving at the conclusions that they are, right? Because sometimes we don't know how they're getting from A to B. We're giving them kind of all of the ingredients, and then a cake comes out at the end, but we don't really know how that happened. And so what he found in the study that he was working on was, and it was a simple model of showing the model images of wolves or dogs, and could it correctly classify that image as a wolf or a dog? And it was doing extremely well at doing this. But what he found was it was not actually predicting that there was a wolf in that picture. It was predicting that there was snow actually in the background of those pictures, because the data that was used to train the models using pictures of wolves, they were often taken in the wintertime where there was snow around. So yeah, the best model of a cat is a cat. Sometimes you have to pay close attention to, is it actually the cat that the model is predicting, right? Because it may not be. I just thought that was really interesting, but thank you.
From Ashley Lynn. How do we connect with the Seeds of Success program? She's part of a group that collects seeds in Central Texas and rescues the creation. Alternatively, or additionally, how do we send seeds to the seed bank at BRIT or obtain a list of target species? Oh,
fantastic. Yeah. So there's one organization in Central Texas that I know actually uses the Seeds of Success protocol actual, and that is Lady Bird Johnson Wildflower Center. However, it's important to note that the Seeds of Success program does not collect state threatened, federally threatened or endangered species as part of their program. That is not their focus. So what we're actually doing here is using an adapted version of that protocol. So we're collecting a lot of the same data and phenology data associated with seed collection with our collections that we make here at BRIT. So my definite advice to you is please reach out to me. My contact information is khankins@fwbg.org. or Brooke Best, @fwbg.org. We would love to give you some of those details about how you could send seed to us and also kind of participate in seed collection efforts of rare and threatened Texas native species here.
Other question was kind of related to that. You answered it. What was are there ways to contribute to citizen science with collecting or cultivating seeds for our own projects? For example, collecting seeds within a prairie remnant at a cemetery and cultivating them for wild landscapes installation in nearby locations.
Yeah, iNaturalist and other citizen science data repositories play a huge role in kind of the availability of occurrence data that we're using to generate a lot of these models. And depending on kind of the application, right? I was not using iNaturalist data to identify mollusk species because it was hard to do. But for the case of like seed collection and cultivation, having citizens update where they're actually observing these species to occur and making collections of regionally adapted seed that you're planning to use in other similar ecozones and ecosystems is a fantastic way of ensuring that the seed that you collect is going to thrive in the areas that you are restoring with that seed. So yeah, I definitely encourage people to use and collect data through whatever means they have available. iNaturalist is a great one. And then the SOS protocols and some of those considerations for safe and mindful collection of native plant seed you can access freely. Just search Seeds of Success. There's a lot of BLM pages that will provide PDFs of the protocol that is used, and they kind of go into a lot of depth about a little bit of what I touched here, but even further about some of those considerations as you're collecting native plant seed that you should definitely check out if you're interested. Yes.
I have a quick comment. I really enjoyed your talk and you and my tips. Your talk make me think wildly about, I work on grapes. And there are like 3 species of grapes in Texas. They're very narrow endemics. I was thinking about the herbarium here has good data for the distribution for the points. And then maybe there's a greater need of using the herbarium collections and look into the future.
Yeah.
Of those species.
Yeah, no, I think that would be awesome and definitely 1000% possible. So yeah, no, if you're interested in looking into any of that, I'd be happy to help, but also like all of the really cool thing about modeling in general is that most of it is open source. And so you're able to use things like R and publicly available data to generate some of these model predictions in ways that aren't cost prohibitive, which we love in science. That's our favorite thing, is things that don't require lots and lots of funding to do, right? So. Mm-hmm.
I wonder if this modeling can point us to the mechanisms, why they are so restricted.
Right.
In comparison to the closely related systems.
Yeah, no, that would be really, really interesting to try to kind of like elucidate whether maybe that's climatic variables. Are there some sort of biological interactions that are going on that are limiting those species distributions? And that's, I think, one of the coolest applications of these techniques is kind of helping you develop some of those hypotheses and give you directions to go in to kind of assess some of those questions. Yeah.
All right, well, that's time here. I want to make sure everybody gets a chance to sneak out gracefully. If you have more questions for Kay or about the seed banking program here, you're welcome to stay after to come chat with us. Thank you all. Thanks, Kay.
Yeah.
Did you ever get to see any of